On today's biocast, we're going to talk about the light-independent reactions, or what's also known as the Calvin cycle. You should have already watched a biocast dealing with the light reactions, and this equation should therefore be familiar to you. It's the equation for photosynthesis. If you remember from the light reactions, what we dealt with there was we dealt with the splitting of our water molecules, we dealt with the photonic energy, and we dealt with the production of some molecular oxygen as waste products. So that leaves only two parts of the equation that we haven't dealt with yet. So that's obviously going to be what we're dealing with in the light-independent reactions. Carbon dioxide that's brought into the plant from the atmosphere, and then the productions of sugars. So let's take a look at the light-independent reactions. Now, the Calvin cycle, or light-independent reactions, is a cycle. It's a recycling system essentially for restoring our major component that we're interested in, which is this 5-carbon 2-phosphate molecule called ribulose bisphosphate. Within the light-independent reactions, we have three main phases: carbon fixation, reduction, and regeneration. And we're going to take a look at each one of those in turn and see how the molecules involved change in the production of that sugar. So we're going to take our ribulose bisphosphate, and we're going to take 3 carbon dioxide molecules that we've brought into the plant. Now, we are only going to follow one of those carbon dioxides. So just remember that everything that you see happening is actually happening in triplicate. We're going to send our ribulose bisphosphate over to interact with that carbon dioxide. What we're going to need to do here is we need to break apart the bonds connecting our oxygen to the carbon, and then we need to form new bonds between the carbon that was associated with the carbon dioxide and the carbons that are associated with our ribulose bisphosphate. In order for us to be able to break apart the covalent bonds in the carbon dioxide and catalyze the bonds between the carbon and the ribulose bisphosphate, we're going to need an enzyme again. And the enzyme we're going to be using is Rubisco. Rubisco is going to pull off those oxygens, and it's going to catalyze the covalent bonding of the carbon to the ribulose bisphosphate. Now, we're done with the Rubisco, so it can go away, and we're left with a 6-carbon 2-phosphate molecule shown here. Now, this 6-carbon 2-phosphate molecule is very short-lived. It's just an intermediate state. It's unstable, and it quickly breaks apart into 2 3-carbon 1-phosphate molecules called 3-phosphoglycerate. So we've essentially finished fixing our carbon. We added the additional carbon from the carbon dioxide to our ribulose bisphosphate, and we have now broken apart that short-lived intermediate into 2 molecules of 3-phosphoglycerate. The next step we're going to do is going to be adding another phosphate onto each one of these 3-phosphoglycerates. For us to catalyze the formation of the bonds between that new phosphate and these carbons, though, we're going to need energy. And we're going to get that energy from some of the ATP that we produced in the light reactions. So let's take a look. We're going to move down our Calvin cycle a little bit. We're going to bring in ATP. It's going to be reduced down to ADP, freeing up a phosphate. That energy that's produced when we break off the phosphate from the ATP is going to be used to catalyze the covalent bonding of this phosphate to our 3-carbon 1-phosphate molecule. And then our ADP will go back towards the light reactions. We'll do that again for our second molecule. ATP comes in, bust off a phosphate, attach a phosphate to our 3-phosphoglycerate molecule, and now this is called 1,3-bisphosphoglycerate. Now we're changing our molecules more in a little more detail than what you're seeing. We're just trying to catch the basics here, and it's easiest to follow the carbons and the phosphates. So that's all we're looking at. So the next step is going to seem kind of odd, because the next thing we're going to do is we're going to remove the phosphates which we just attached. It's going to take energy for us to break those phosphates off, so we're going to bring in another one of the molecules which we produced in the light reactions, which was NADPH. The NADPH is going to take the energy associated with it from the attachment of that hydrogen, the H, and we're going to pull that off, converting it to NADP+. The energy that we gained when we broke off that hydrogen, we're going to use to separate the phosphate from the 1,3-bisphosphoglycerate, and we're going to do that again to our second molecule. NADPH comes in, we convert it to NADP+, that gives off energy. That energy allows us to remove the phosphate from the 1,3-bisphosphoglycerate molecule, and now we have what is called G3P. The G3P is essentially what we were trying to get at. One of these two G3P molecules is going to be used to build glucose or other organic compounds. The other G3P molecule is going to be used to regenerate our ribulose bisphosphate molecule. So we're going to take and we're going to send one of those G3P molecules out to wherever the plant needs it. The other one is going to remain in the light-independent reactions, staying part of the Calvin cycle. So now we have a G3P that is being utilized by the plant to build sugars, which is our C6H12O6. And then we have another G3P molecule, which is going to be used to regenerate our ribulose bisphosphate. Now, for us to add additional atoms to this G3P molecule, we're going to need energy, and we're going to get that energy in the form of ATP again. We're going to take and have ATP generate the energy that's going to catalyze the covalent bonding of a phosphate and 2 carbons onto our G3P molecule, which you'll notice has recreated a 5-carbon 2-phosphate molecule, which is ribulose bisphosphate again. Now we can start the whole process over again with our ribulose bisphosphate, a new set of 3 carbon dioxide molecules coming into the plant. We bring our ribulose bisphosphate over there. We bring our Rubisco in. The Rubisco is going to catalyze the breaking of the covalent bonds between the carbon and the oxygens. And then the formation of the covalent bonds between those 2 carbons. So now we have made it through the entire portion of the photosynthesis equation. We had water molecules and energy being utilized in the light reactions, which ended up producing molecular oxygen waste, as well as the NADPH and ATP, which we used in the light independent reactions. And then in the light independent reactions, we had our carbon dioxide being transformed into sugars and other organic compounds that the plant needed. So I hope this helps.